afternoon and good morning or good evening, everyone, depending on where are you joining us from. For those who are here for the first time, my name is Andrea. I am based at the University of Tübingen, and it is my honor to be hosting you today on a new session of our webinar series for the Africa-Europe Cluster of Research Excellence on Advanced Infectious Diseases. As it's our routine for every month, we are introducing more members of the CoRE-AID network. For today, we have Professor Steffen Borrmann at the University of Tübingen and Jonas Luginbühl, I hope I'm pronouncing it correctly, at the University of Bern. And for today, it is also my pleasure to reintroduce Professor Bismarck Dinko. Bismarck is a senior lecturer and a group leader at the Kwame Nkrumah University of Science and Technology at Kumasi, Ghana, but at the moment he's doing his Humboldt Fellowship at the Heidelberg University Hospital, where he works on malaria and bone marrow and adaptations of malaria to different tissues. With this, I will hand it over to Bismarck, and I really hope that you enjoy the session of today.
Yeah, thank you very much, Andrea. And I will go straight to the point to introduce the first speaker. So the first speaker for today's webinar is Professor Steffen Borrmann. And Steffen is a professor of tropical medicine at the Institute of Tropical Medicine at the University of Tübingen. He got his medical degree and training several years ago, and then he's interested in the development of novel interventions against malaria and other infectious diseases. And he's currently involved in several multi-center projects. And then he leads and participates in these projects as a principal investigator. At the moment, he's focused on implementing a first in Africa controlled human infection using local Plasmodium falciparum infected mosquitoes in a new platform. And today he will talk to us about a new controlled human malaria infection model using Anopheles gambiae. Professor Steffen Borrmann, please.
Thanks, Bismarck, for the nice introduction. Thanks, Andrea, for organizing and hosting this. Can you hear me?
Yes.
Super. Can you see the slides?
Yes.
Yes, we can see the first slide. Yeah.
Okay. Super. So I have to start with a disclaimer. So this is a work I'm going to present today, which is not published. So please refrain from distributing this. So it's still unpublished, one thing. And the second thing is, this is, of course, as you may imagine, the work of a large team, but also, and not also, but mainly the work of a PhD student, Anik Barra, who is also present here today. Hi, Anik. Thanks for joining us today here. So I'm going to track you to Gabon before we return to Europe, namely Bern. And I'm not going to introduce you to the life cycle of the malaria parasites, but just to emphasize here very quickly that numbers do matter in the life cycle. So we have these famous bottlenecks that typically occur during the host switches from the mosquito vector to the human host and back to the mosquito vector, and they're interrupted by these massive replication phases occurring both in the mosquito vector but in two different stages in the human host, namely liver stage and the asexual parasite stages, and the numbers are displayed here, and they're massively different. We can observe up to 10 to the power of 10, 12 or so in human infections in the blood stages. However, far fewer parasites are being injected by a mosquito. Typically, we think less than 100 per mosquito bite. Far fewer than infect hepatocytes, but in a single round of replication within 1 week, in the case of falciparum, up to 10,000 daughter cells are emerging, are being released in the blood stage per sporozoite. So And the second thing important for us here, because I'm going to mention this, and also as a rationale for us to establish this human infection model, is the fact that we see substantially different vaccine responses. For instance, if we use sporozoites that are being transmitted, and infections in the liver stage are either abrogated very early in the liver stage, where we are talking about a few hundreds or even thousands, or in vaccine studies, many thousands of parasites, but before replication, importantly, and vaccine efficacy is hugely inferior to compared to when we allow parasite replication in this stage to the end full maturation after 7 days with 1,000 to 10,000 more parasites that have been developed and differentiated during this 1 week of development in the liver stage. I come back to this and also towards the end to try to point out why our model system could be useful for future directions, malaria vaccine development. However, at the moment, we face a dilemma, and the dilemma is that we have a whole parasite vaccine approach that works 100%. For instance, in Tübingen, volunteers, exemplified by a study published in 2017 on the left-hand side, where you see at the look at the Kaplan-Meier curve after challenge, after free immunization, and all controls. Here's the gray curve become positive by thick blood smear. This is not qPCR data here. This is a microscopy here. However, all vaccinees remain negative after challenge. If you do the same thing and even increase the dose to 100,000 sporozoites attenuated by chloroquine and free immunizations with a challenge roughly at the same time, but the same challenge, importantly, in semi-immune adults in Equatorial Guinea, you have far less protection. First of all, you are failing to infect all of your volunteers. And secondly, vaccine protection, as exemplified in the reverse Kaplan-Meier plot on the right-hand side, is largely substantially inferior to what we have achieved in non-immune volunteers participating in our study, for instance, in Tübingen, but also in other places. So this is well established, and it's a problem for us because we would like to develop a vaccine that doesn't work in non-endemic areas. We need a vaccine that works in endemic areas. There are different methods for conducting controlled human malaria infections. Very quickly, there are two methods that use sporozoites here. One is by mosquito bite transmission, one is by syringe transmission. And of course, there's a more exotic method to induce controlled human malaria infections using blood stage parasites, which is rarely being done. But historically also, mosquito infections were done for many decades. More recently, through development at Sanaria, an US-based company, which succeeded in making cryopreserved aseptic sporozoites available that can be inoculated via syringes. We made big progress here, and this was also used in many study sites. However, we went back to the mosquito-transmitted model for a few reasons, but most importantly for us, we think that provides us the flexibility for testing new interventions and new protocols, but also in research questions that would be otherwise difficult to address using a GMP-grade sporozoite formulation. Few quick further words for the background to our study. So essentially, of course, if you use mosquitoes, in our case, Anopheles gambiae mosquitoes, which is the main vector for falciparum on the African continent, plus by infecting mosquitoes with locally circulating parasite isolates, We are getting very close to achieve something that what we call a controlled natural transmission setting, since at least the mosquito, even though it's a Kisumu strain, it's the main vector of malaria parasites. And again, we're using naturally circulating parasite isolates to infect those mosquitoes. So this is quite far away from using Anopheles defensii for these infection studies in European countries, for instance, but also using lab strains that have been dragged into culture for several decades. So we think this may be important, and I hope I can convince you that indeed we are able to achieve infection rates that may have been difficult to achieve with lab strains and the typical mosquito bite controlled human malaria infection model. Of course, there are a few key requirements that you need if you want to establish such a platform. We need an infrastructure that has containment for the infected mosquitoes, and we need to be able to show that we have high and consistent salivary gland infections in all mosquitoes. These are a few pictures from the infrastructure that was built and established quite a few years ago. And then the team was able to use this infrastructure after many refinements to achieve high infection rates in our mosquito colonies. These are exemplary data here, both looking at sporozoid prevalence rates in our mosquito colonies, as well as sporozoid intensity in the mosquito colonies. And with these things and a few other things, as you may imagine, since it's a clinical trial, we designed a first study where we intended to use 5 infected mosquito bites for the inoculation. A quick word for the 5 bites. This is a gold standard. Of course, people have achieved success rates, infection rates, close to 10 out of 10 with lower numbers. But generally accepted is 5 infected mosquito bites, so that's what we started. But as you can see here, we basically assumed that these 5 infected mosquito bites would not be sufficient to infect 10 out of 10 volunteers, since, as pointed out earlier, typically in semi-immune populations, infection rates are expected to be lower. So we designed a study where we would go on and increase the number of mosquito bites by the factor of 2 to 10 bites if infection rates would be below 50%. Importantly, of course, for safety, we had quite a few hard criteria typical for these studies, where during the follow-up we had daily qPCR from day 5 to day 21, and we had several criteria to put in place to ensure the safety of the patient for initiating treatment, as indicated. For this study, we have 2 different lines of operations. One is the clinical team. The clinical team in screening volunteers, initiating treatment. In this case, we use clindamycin for 5 days, 300 milligrams twice per day. 7 days before the CHMI day, and then there was a follow-up in our cohort. At the same time, the parasitology entomology team infected mosquito colonies. This is also a kind of 2 weeks process where, first of all, the donor had to be identified, colony had to be infected, and there was a second non-infected mosquito feed 7 days after infecting the colony. And at the same time, there were several tests done for the safety of our procedure. Specifically, we had to ensure that we are not transmitting multi-drug resistant parasites here. So ex vivo drug assays were conducted. But we also had to rule out 2 Anopheles mosquito, potentially Anopheles mosquito transmitted pathogens, onion young virus, and Vocaria bancrofti, which was excluded by qPCR. So for our donor, we had a few criteria here. Of course, we screened healthy donors, but we needed gametocytes clearly to infect mosquitoes. Also asexual parasites, not a minor thing because we need to do ex vivo drug sensitivity assays, so gametocytes alone would not be sufficient. As I mentioned before, the donor blood would be screened for these two potentially Anopheles mosquito-transmitted human pathogens. Drug assays were conducted, and altogether this was forming what we would call a release criteria before and a batch of infected mosquitoes could be used for the CHMI in our study. As I mentioned before, we started with 5 infected mosquito bites using a set of standard operating procedures that were developed in a nice collaboration with Radboud University in Nijmegen, a partner here in our initiative, of course, as you know. Not so sure whether we have colleagues today from Radboud present. I hope so. And we also were able to access standard operating procedure from Sanaria, the before mentioned American company that was succeeded in producing aseptic cryopreserved sporozoites for these CHMI studies. So altogether, a very nice collaboration. Without this, CHMI would not have been possible. And of course, many colleagues in the team. Again, coming a bit, extending a little bit on the follow-up here because it's specific, because this is done in an endemic area where natural reinfections could occur. So we advocated the use of personal repellents, bed nets, and of course we implemented genotyping for distinguishing potential natural reinfections from our CHMI infections. If you think of a mosquito colony that is infected to 80%, and you need only 5 infected mosquito bites, you probably would guess that we need very few mosquito bite attempts to infect people. But as you can see here very easily, that for some volunteers, up to 4 attempts were needed to make sure that we have always mosquitoes that were blood fed. If they were not blood fed, they were of course replaced, and if they had less than 10 sporozoites, they also had to be replaced. So altogether, sometimes 4 rounds of these exposures had to be conducted by the team. So jumping straight to the results here. We had 2 cohorts. So this is the data from the first cohorts. 10 volunteers were exposed. This is a qPCR data that you're looking at. On the y-axis, parasite T-mails are expressed per milliliter, and on the x-axis, the time by days. By starting on the CHMI day 0. First observation is there are some individuals, some study participants who were who still carried submicroscopic parasites before infection. However, and generally, the study was highly successful because we have succeeded in infecting 10 out of 10 individuals. Even though, as you can see already in the first cohort, I hope you can see my error here. Andreas, it's possible. Maybe I can also use the... So as you can see, for instance, this individual here with a relatively high submicroscopic parasitemia who hesitated, but then finally also became positives. We can confirm that these were indeed parasites that were inoculated by our CHMI, and they were hence they were not due to natural reinfections in our cohort. These are the MSP2 genotypes. We had other genotypes, MSP1 and 1 microcyclide marker, but here we look at MSP2 because it typically provides very nice genetic diversity here. In red here, these are the different genotypes observed at that locus in the isolate obtained from the donor. Then in green, this is data from the sporozoites. This is, of course, already after meiosis that occurs in the midgut of the malaria parasite. So representing definitely, we think, different clones, but the same genotypes sitting probably on different clones after recombination. And in blue here, you can see genotypes observed in the 10 volunteers. I think it's a clear picture where the dominant genotypes in most cases were nicely being transmitted to our volunteers. So with this, of course, and according to the study design, we went ahead with a second cohort, again with 5 mosquito bites, which was also quite efficient or even more efficient than the first cohort because only 65 mosquitoes in total were needed to infect 10 volunteers. 50 would be the minimum, so 65 is a very efficient figure. And this is the data, again, qPCR data from the second cohort, y-axis again, representing numbers of parasites per milliliter, and on the x-axis now, the qPCR data by study day. You can see that for the second cohort, we started with the qPCR a bit earlier, in fact, pre-treatment, since we had this observation that some who did not clear parasitemia had some delay in developing parasitemia. So we are wondering whether pre-treatment, whether or not our volunteers who were required to have a negative thick blood smear. So this is all submicroscopic infections. But you can quite nicely see here that quite a few, if not, the majority of our volunteers had clearly evidence for submicroscopic parasitemia before starting off treatment. For some, parasites seem to have been cleared, but not for all. In fact, there were these 3 individuals here who had some decline, but nonetheless seemed to persist despite a supervised 5 days of clindamycin, which has activity not just against asexual parasites, but also against liver stages. Interestingly enough, in genotyping, again, MSP2 data here, this seemed to have been a single clonal infection, so at least one genotype. We don't know whether this genotype exists on several clones, but we assume it's a single clone. And we can see nicely here that this clone or genotype at least was nicely transmitted to mosquitoes and then to our volunteers and had been recovered from our breakthrough blood stage infection. So in the second cohort, we had 9 out of 10 individuals infected. I would point out here, there was this one individual who was not infected. So altogether, we succeeded to infect in these 2 cohorts in total 19 out of 20 individuals, which we think is definitely exceeding what we had expected from our study. Most of our colleagues would have expected, given that the dose is a dose that we would need also to infect 10 out of 10 non-immune volunteers. So already coming to the conclusion here, because time is running out. Bismarck is already looking sharply at me. So in conclusion, we observe excellent transmissibility of our genotypes. Also, we have high initial blood stage inocular, which indicates high late liver stage parasite loads. And the second observation that we take away from this is that we seem to have difficulties in super infecting individuals with pre-existing persistent blood stage infection, for which we have two leading hypotheses here, and then I'm finishing, Bismarck, with a few words for the acknowledgements. So one thing that Annick is currently exploring or has already started to explore nicely is to see whether this correlates with immunity, and namely looking at immunity against merozoites. We also look at anti-infected red blood cell surface immunity, which is also ongoing. I can't share the data here. We also looked at possible parasite-mediated factors here that could explain this phenomenon. So far, we have explored hepcidin, which was previously identified as correlating with this phenotype in mice, but not sharing the data here today. But trust me that there is nothing to report here for hepcidin. So hepcidin levels did not change or correlate with our CHMI outcomes or the dynamics of our parasite dynamics after release from the liver stage following CHMI. Big thanks to the team. Annick is our PhD student next to Giardia, I think is also present here, and many other colleagues here. Of course, I need to highlight the PI of this project, Jean-Claude Desjons, who led the clinical team here. And we also got some important help from Pete Billingsley, who previously participated in many of these CHMI studies based at Sanaria. And many collaborators, including in Nijmegen, in the UK and the US, as well as in Hamburg. Thank you so much.
Thank you very much, Steffen. It is now time for questions. And then there are lots of hands up, so, okay. Yeah, okay, so let me...
Jonas, maybe, I don't know.
Yes, so Jonas, you can go ahead. Your hand was up.
Sorry, me? I didn't put my hand up.
Okay, let me check. Okay,
Sorry.
so I have a question for you, which is that while we wait, so the experiment makes use of, if I got it right, you have naive individuals and also semi-immune individuals, right?
Yeah.
With respect to the semi-immune individuals, because the acquisition of malaria immunity in those volunteers, malaria immunity is a continuum. Did you profile their immune response to previous malaria infections before they were involved in the trial?
Good point. Thanks, Bismarck. No, we did not formally. Of course, Annick has generated data from these anti-merozoite assays, and there will be further data from the anti-infected red blood cell surface immunity, but we have not looked into this in detail. But we think these are 18 to 45-year-old semi-immune Gabonese volunteers living in a perennial malaria transmission situation. And anyway, we saw the data. All of them, basically all had persistent infections when they entered the study, submicroscopic infections, so they were clearly exposed.
Okay. How about the naive individuals? Is this just based on the information you collect from them that says, okay, we never had came into contact with malaria, or you did some other tests?
Oh, this may be a misunderstanding. So I was referring to studies that were done in students in Tübingen, for instance, who we assume are really non-immune, have never spent time in malaria endemic areas. So this is what I'm referring to. But in this study, only adult semi-immune healthy volunteers from Lambaréné participated.
Right. Yeah, I don't see any questions. Okay, that's a question now. Yeah. Does co-infection, this question is from Francis, does co-infection influence the MSP2 clonal types and immune response?
So whether co-infections?
I assume co-infection with other malaria parasites or other parasites. I think
I assume yes too. Maybe co-infections, others malaria, human malaria species. No, I don't think so. For us, MSP2, of course, is not a neutral marker. That's correct. So it is under immune diversifying selection pressure. It's not the best marker for genotyping, indeed. But I don't think there's a correlation between MSP2 clones or genotypes and immune response. By the way, they don't look into MSP2 immune responses, not yet.
Yeah, I don't see any hand. I'm just checking now. Otherwise, I have to ask.
Mm-hmm. Then thanks again.
Do we have any more questions? Yeah. Maybe later when we get to the end, there will be additional questions. Yeah. All right, Jonas, are you ready? Can you start sharing your slides and then I get to introduce? Okay.
Yes.
All right.
Okay.
So
very good.
Our next, yeah, I can see the slides. Our next speaker for today's webinar is Jonas Luginbühl from the University of Bern in Switzerland. Jonas did his bachelor's and master's degree in molecular life sciences at the University of Bern, and which he completed in 2020, looking at Pee-wee proteins in Trypanosoma brucei. Then he worked in quality control in a biochemistry laboratory for 2 years, after which he got bored, and then he came back to the University of Bern to do his PhD. And today he will be talking to us about his PhD project working on Clathrin-mediated endocytosis in Giardia. Jonas, please.
Thank you very much, Bismarck. Yes, so I will jump right in. Thank you very much for this opportunity to present here this webinar. I noticed I have quite a few slides and not as much time, so I will just go ahead. First of all, I would like to introduce you all to our model organism, Giardia lamblia. As you can see from this video, there are a few key things that I would like to highlight. First of all, when looking at them through the microscope, they do look quite adorable. Secondly, they are motile. And thirdly, they can also attach themselves very well to surfaces. So this is the disease-causing trophozoite stage that you see here, which colonizes mammalian midgut and attach themselves to the epithelium there. What I personally am interested in is how these cells feed so that they are able to divide and do that. For that, these parasites have these vacuolar organelles that sit in the cell periphery. We like to call them peripheral endocytic compartments, or PECs for short. So how do we know that these are in fact endocytic organelles? When you feed these parasites with any sort of fluorescent dye, these are the compartments that you stain. When we take a closer look, so this is a 3D reconstruction done using data generated by super resolution microscopy, you can see that these organelles are in fact polymorphic, so different colors here represent different morphologies. And when we think of them as endocytic organelles, As a eukaryotic unicellular parasite, one of the main pathways that we need to think about is Clathrin-mediated endocytosis. So previous colleagues, when investigating these organelles, have looked at Clathrin in Giardia, and you can see here on the left side that Clathrin in Giardia, in fact, also localizes in the same vicinity as these organelles. Now I need to give you a brief refresher on how Clathrin-mediated endocytosis works. So all of this data is from other systems, mammalian cells and yeast mostly. CME, for short, is named after the main protein player, which is Clathrin. Clathrin comes in this very cool triskelion shape, which just means three-legged, and each unit is made up of 3 Clathrin heavy chains forming those legs, with each of them having a Clathrin light chain attached here. In terms of domains, Clathrin heavy chain has a trimerization domain in the C terminus. It has a light chain binding domain. These pieces together are called Clathrin hub. And then, very importantly, they also have an N-terminal adapter protein binding domain. A key feature of Clathrin is that it's very good at assembling into higher order structures, such as Clathrin cages, just in solution under the right conditions, or membrane coats inside of cells. So how CME looks like in standard textbook scenarios, you have your membrane-bound cargo, which is recognized by the adapter proteins, which leads to the recruitment of the Clathrin coat. This in turn leads to membrane invagination, and then through the action of actin and scission proteins, this vesicle is pinched off, goes on to be uncoated, and deliver its cargo to the endosome. I want to highlight key players here. This is the AP2 adapter complex, the Clathrin in the coat, and dynamin for the scission protein here. Okay, moving back to Giardia. When we take a closer look at where Clathrin sits in Giardia, previous colleagues have done this using electron microscopy. They used a construct that they were able to stain in EM, so you can see these dark blobs. They sit in between those organelles, the PEGs, and the plasma membrane. To further investigate the role of these Clathrin assemblies, they of course wanted to know which other members, which other proteins sit in this vicinity. So they did interactome studies, and indeed they could find some conserved CME proteins. So I just mentioned the AP2 adapter complex. They found subunits of those. They found a dynamin-related protein, as well as some phospholipid binding proteins. Phospholipid binding proteins, such as this PX domain protein here, were also shown to play regulatory roles in CME in human cells. I mentioned PXE2 here because it will come back later. Another interesting protein they found was this previously unknown ORF4259, which, when they looked at it more closely, thought to be a Clathrin light chain analog. This was hypothesized because it is the most stable and most abundant interactor of Clathrin heavy chain in Giardia. Also, it matches the uniquely static behavior of Clathrin heavy chain in Giardia. And when you look at its predicted structure, they saw that it, like other light chains, has a central alpha helical rod, a C-terminal helix-terminal helix motif, and an unstructured region in the N-terminus. However, on a sequence level, it is not identifiable as a bona fide Clathrin light chain. At this point is where I started my PhD. So knowing all this, I formulated my central hypothesis, which is that Giardia's Clathrin assemblies play an essential role in molecular trafficking into the cell, and they use an evolutionarily unrelated structural and functional analog of a Clathrin light chain to do so. I formulated several sub-aims to test this hypothesis, to untangle it a little bit. So first of all, I want to investigate the structural configuration of these Clathrin assemblies. I want to investigate their function in endocytosis, and I want to investigate the state of divergence of Giardia's Clathrin in relation to other organisms' Clathrin. In the talk today, I will present to you 3 pieces of data, one from each of those sub-aims. So I will present to you some cross-linking mass spectrometry data. For the structure part, I will tell you about truncation experiments and also a functional complementation experiment putting Giardia Clathrin into yeast. Starting with the cross-linking mass spectrometry. So this is a method which tells us which folds of a protein complex were in close spatial proximity in our sample. It does that by using a cross-linker and then followed up by MS analysis. So we find out which peptides were cross-linked, again giving us the information of which folds of a protein complex were in close spatial proximity. Just to remind you of the Clathrin structure again. So if we expect Giardia Clathrin to behave like this, we could expect cross-links from the trimerization region, or for example, also from the light chain analog binding in this region here. And I'm going to move straight to the data. So this is a representation of my cross-linking data. You can see, so each of these bars represents a protein that was cross-linked to Giardia's Clathrin. We have the heavy chain, of course, we have the light chain analog, and we have this PX domain protein too. For now, I would just like you to focus on these. Should mention also like all these gray lines are individual crosslinks, and I would like us to focus just on these crosslinks in between the Clathrin heavy chain and the light chain analog, because when we map them onto an AlphaFold predicted structures, we can see first of all that the AlphaFold predicted structure agrees with the data I generated, and also the binding site, the binding region, the footprint of Giardia light chain analog and Giardia heavy chain binding site is the same as it is in bona fide Clathrin. I think what's pretty convincing there is that we have a cross-link N-terminally of this alpha helical rod structure, as well as several cross-links C-terminally. So this leads me to conclude that the heavy chain light chain footprint is conserved in Giardia despite this highly diverged light chain analog. Moving on to my experiments with Clathrin truncations in Giardia. So the rationale for these experiments was to chop off individual domains of Giardia Clathrin heavy chain and see, when expressing them inside a cell, see whether the localization changes for any of them, thinking when I mess up anything important, this very distinct localization in the periphery is likely to be messed up as well. So first of all, I show you the full length control where I removed no domains. Then a truncation where I just removed the trimerization domain. Here again, the localization is not impacted. When I just express the hub region that I've mentioned before, we still have peripheral localization. However, there is some punctate localization in the cytosol as well. And when I now remove the trimerization domain from this piece, however, the peripheral localization is lost completely, and also protein levels of this construct are much lower. So to me, this tells that there is a role played by the trimerization domain also in Giardia's Clathrin. I should mention here as well that this is ectopic overexpression of constructs in a wild type background. So we can always expect there to be piggybacking effects of the truncated versions of Clathrin with its own full length. I did then also CoRE-AID MS experiments to see whether they still interact with the members of their core complex. And this showed me that in this final construct, these interactions were lost. Also, I got high levels of ubiquitin in this MS sample. So I conclude from that this construct is being actively degraded. Moving on to the complementation experiments in yeast, since my time is also already running short. I'm not going to go into detail about how my system really works. I just want you to know that when so what happens is I knock down the endogenous version of Clathrin in yeast. In that case, yeast loses the ability to grow. In the other for the experiments, I add back another version of the protein that can be either yeast's own Clathrin or Giardia's Clathrin, and I hope to see growth being restored if that function is replaced. So showing the data briefly here. So we have the wild type case, so no Clathrin is being knocked down, the yeast are growing normally. In our knockdown controls, the yeast cannot grow since they lack the Clathrin. When we add back the yeast Clathrin, the growth is restored even to a slightly higher level than our positive control. Just adding Giardia Clathrin does not aid them. However, we can express a chimera. In this chimera possesses the hub region of Giardia Clathrin and the rest of yeast Clathrin. And interestingly enough, this chimera does complement the growth phenotype back to a wild type level, telling us that this part of the protein of Giardia can functionally replace yeast's hub domain. I also conclude from this data that very likely trimerization is occurring in Giardia, and that it can happen independent of its own light chain because, of course, the yeast that we use don't express Giardia's light chain analog. Now, this leads me to my summary. I hope I could convince you that the bona fide heavy chain light chain footprint is conserved in Giardia, that trimerization seems to play a role based on the last two pieces of data. And to conclude it all, I would just, we can see here that despite Giardia's divergence, the very basic assembly principles of Clathrin complexes are conserved. Thank you very much for your attention. I also would like to say that data that I showed here was generated in part by Alina, a master's student in our lab, and also in collaboration with Selina, a bachelor student that worked previously in our lab. Thank you very much.
Thank you very much, Jonas. Now it's time for questions. Yeah, so Carmen's hand is up. Am I right? Please go ahead.
I'm sorry, were you speaking to me, Bismarck?
Yes, I saw that.
No, I was clapping. I was congratulating
okay,
Jonas.
okay.
Sorry.
Oh yeah, okay, I see another clap.
I can grill him any day in the lab. I'll leave it up to others to grill him. It's fine. Not my job today. I'm just listening and enjoying.
Okay, Stefan too is clapping. Okay. So yeah, while we wait in the yeast experiment,
yes.
Is it only when you knock out the Clathrin protein, is it only the growth defect that you see?
Of course, we could also design experiments to directly look at the endocytic uptake. But it's much easier for us to just look at the growth because you can see a very, very clear phenotype there. So this is much cleaner for us.
Yes, actually, you answered my second question. I was going to ask whether when you do the complementation, does the protein function as it is expected? Yeah, but you've answered that. Yeah, so there are experiments that you are yet to do. Okay. Okay. Yeah, I don't see any other hand.
Very much a change of pace from the very clinical first talk to the very molecular second one.
Yes, yeah. And how similar is the Clathrin assembly in Giardia compared to malaria parasites? Do you know?
I haven't looked at
malaria. Okay.
At malaria. No, I don't actually know the CME system in malaria at all. Sorry.
Okay. All right.
Steffen has a question.
Let me check.
Yeah.
Yes, please go ahead.
Thanks, Jonas. Nice work. Interesting. So let me give it a try for a question, since you mentioned it's difficult to bridge the lab and the clinic sometimes. So you can cue the question, of course, which is, are there any known inhibitors for Clathrin as a target of intervention? Any chemicals, scaffolds, etc., something in the pipeline for any other?
I mean, I didn't check. I would be surprised. I mean, it's of course it's a very, I mean, it's conserved across eukaryotes, right? It's a very basic pathway, and so in that sense, I would be surprised if it were possible to use it as a therapeutic target without risking effects to the patient itself. Also, I mean, it's just in the case of Giardia, it is quite far diverged on a sequence level from many other organisms. So I mean, I'm thinking like in terms of my own research here, even if there was anything, it's often it's often the case that these drugs, these molecules, then might not work in Giardia itself due to the high divergence. Yeah.
Yeah.
Kwame wants to
grill you now.
Yeah, actually, I 100% agree with what Jonas said, and I have two comments. The first one is, Bismarck, the question you asked concerning CME and Plasmodium is actually a really interesting one, because we know that the adaptoprotein complex in Plasmodium, in the apicomplexa in general, has, from a functional perspective, diverged quite significantly. So it's really interesting, in fact, to see how this universally conserved process has taken on different looks in different lineages of protist parasites. So I just wanted to point out that that's actually a really interesting and intricate question. And concerning what Jonas just said, 100% agree. What might work is working indirectly on Clathrin-mediated endocytosis. So, for example, an antibiotic that we use to select for transfectants in the lab is neomycin. And neomycin actually targets the metabolism of lipid residues that are important for Clathrin-mediated or for Clathrin assemblies to be anchored to the inner leaflet of the plasma membrane. So neomycin itself can be used as an inhibitor for the turnover of these lipid residues that mark sites of endocytosis. And it is very toxic to Giardia cells, as it would be to any cell that has active Clathrin-mediated endocytosis, because it interferes with the lipid-specific marking of endocytic sites. So acting directly on endocytosis might be a difficult thing to do, but acting indirectly with something that interferes with the process but doesn't kill it altogether might be an interesting way of thinking about this.
Great. All right. I don't see any more questions. So if there are no questions, Andrea, back to you.
Thank you, Bismarck. Thank you again to our speakers, to Steffen and Jonas for the talks, and everyone for participating. I just want to give you a heads up that we will go on a summer break, so we won't have a webinar in August. we will meet again on the 1st of September. So I hope you have a nice rest of the afternoon, and we'll see you again in 2 months' time. Thank you very much, and bye.
Bye-bye.
All right.
Happy
Bye.
break, everybody.
Thank you. Bye-bye.
Bye.
Thank you.